Okay. So I think we're ready to start here, and so I'm going to go ahead and get us going here, even if it's a couple of minutes early, because there's always going to be, we could always have a longer break or something. So I want to talk about a project, kind of what the deliverables are. So we have, and then kind of work into the bioinformatics and be able to pull up some actual some data that we did here that we can look at. But I just want to go through the whole thing. So we had talked yesterday about having the consult on the front end and having an experimental design and being able to have a sample list that's filled out and all of that. And I just want to say that one of the things that Sam does before we start any project is we want to make sure that you, as a PI and the resource, that we're all communicating clearly. So every project is going to have a project summary that is going to have an identifier, and then it's going to basically indicate the sample types and the number of samples in the organism. That's really important information up there. And one of the things that Rick brought up yesterday that I wanted to reinforce is that some of these things that we're telling you that are needed or necessary for proteomics are it's not just for our place. It's like any place that you would, any place that you would go, you would have to have essentially similar requirements to do proteomics. So besides in the project summary there, you've got a little summary of the project, and that is always something that Sam kind of puts together where after talking to you, sometimes this is something that you've put in an email where you have essentially given a word, a sentence or two description of what your experiment is about. And sometimes that language means very little to us, right? So I did this on purpose. I've got this thing, and this means a lot to me. I've got these two conditions, staggered and non-staggered. And you're like, I don't have any idea what that is. And a lot of times when you guys say that you've got this treatment or whatever, you've got this certain condition thing, we're just hoping that you understand your biology. What the important point is that we know that you have some separate groups here, and we know which replicates go into which groups and which groups you're going to compare, right? So all of that other, I'm going to call it biological jargon and stuff, a lot of times can be almost a distraction to us, because what we really want to know is that you have groups A and B and C and D, and you want to compare A and B and C and D, but not A and D or whatever. That would be helpful to us. So to kind of put that into sort of layman's terms, one of the things we put is the number of experimental conditions and the number of replicates there. Then we have a project workflow there where we basically say, this is what we agreed to do with your samples when you send them to us. And then down below here, again, what the actual sample groups are, and then what those comparisons that we're going to have. That's really important there. And then, of course, some information down there. And you'll notice we are in line with everybody else in the country and the world. There's fine print at the bottom of our project summary there. So all of that stuff. And then there's an estimated completion date. And that is, like I said, our best given instruments that work right, supply chain issues, people going on vacation and holidays, and the academic calendar and all sorts of, depending on how many samples we have. But we do try our best to try to get all that done in that amount of time. So then the sample list, and Karen talked about this a little bit yesterday, as well as Sam did as well. But just making sure that you have sample names and sample groups identified, the organism, and then the sample buffer is always really key. And what I did down here is I put a key down here of what the comparisons I had, and I just made them A and B. Sometimes you guys, I say you, I'm just lumping all of the PIs in the world together. You will have these really complicated, it's like a certain cell type with a certain condition with a mutant in there. And so you've got these names that are like 48 characters long with all of these special characters in there and all that kind of stuff. And you're doing that, right? Don't give me that look. And so sometimes it's just easy for us to be able to kind of simplify that down. Now, one of the things we need to do when we do our downstream bioinformatic analysis is we need to make a metadata file, and that metadata file comes specifically from our sample list. And you'll notice that there's some extra things in here, and that would be, basically we want to identify the groups over there. And then the other thing that we do besides a metadata file is we've got a contrast file. This contrast file is where we indicate in which groups are going to be compared. And you can see there's a language here. And the important part is that the first part that has the HeLa B versus HeLa A, that is basically what the title is going to be for that particular volcano plot contrast. And then the equal sign, that's actually what it's going to do. Mathematically, it's going to take the B group and subtract from it the A group. And that's really important because sometimes you'll have some, you want to know if this protein has been upregulated, it's been upregulated in relationship to what? And so sometimes we have to kind of guess what the control groups are. Sometimes you guys are adding a treatment that is supposed to bring you back to homeostasis or something. And so you ask for things to be done in a way that doesn't quite make sense to us. And so it's really important that you define all of that so that when you see this protein is upregulated, it's upregulated in the relationship that you were thinking about. There wasn't too many years ago, there was somebody that there was a whole bunch of scientists that thought that there was one particular scientist in their field that had basically had done their experiment wrong and was basically pushing a drug treatment that was essentially pushing the biology in the exact opposite direction. And it took a whole cohort of people to kind of bring that to a stop. So we don't want to do that. We want to make sure all that is crystal clear what we're doing. So when we get that data, we're going to search it against the library. So we have the collected data. We're assuming at this point that we have collected the data off of the mass spectrometers. We know the groups that are to be compared, and that we have a FAFSA library that we're going to use. Some Uniprot, it'll almost always be a Uniprot library that we will use. And unless you tell us otherwise, if you give us a specific library, otherwise we're going to go find one for that particular organism. And then we use a search program to match spectra from our data to spectra in the library from the organism of interest to determine which peptides are in the sample. And when we do that, I think Kevin did a great job yesterday at lunch talking about some of the different products that he was comparing, Diane and Spectronauts and Proteome Discover and those sorts of things. Those are all different searching, database searching programs here. And so that's sort of the starting point for bioinformatics, right? The bioinformaticians sitting around, the samples have come in, they're biologically frozen, frozen samples, and the bioinformaticists are going waiting, and then all of that gets turned into peptides, and suddenly there's data collection, and they're still waiting, and now finally those raw files are there, and they're like, okay, now we can do a database search, and we can start to work. And what they will do is do that search program, and it will give us an output file. So when you get output from us, you will get an output that has a list of, if you're doing a DIA project and you have I guess I should probably use a pointer or something here. So you would get a folder like this that has your conditional setup, some other files in here, but one of them is called a protein report, and it's a TSV, that's a tab separated value folder that you can open up, file that you can open up in Excel. And that is the raw, unnormalized data, right, that has come from our database search. It's not very pretty, and it looks like this, but it's really broken down into two parts. You've got the annotation data over here that is, again, coming from our library. It tells us what protein groups and gene names and what the organism is, and gives the descriptor and all that kind of stuff. And then there's some other information that we can download other columns. Sometimes we're interested in knowing. But for the bioinformatics, we really want that annotation information, and then we get the MS2 intensities, these values here. So each one of these columns represents a sample, and each one of these numbers represents an intensity for the protein that we have in this particular row. And you can see that there are numbers in here, and then there's some that say NaN. That is a spectronaut artifact. They use NaN. Some say Na. Some will actually just put a 0 in there. But bioinformaticians are a little bit picky about that, because 0 means that it's been measured and the quantity is 0. Na means that
Not a number.
It's not a number. We don't know if it's there or not. We just know that we didn't measure it. So it can be absent or it can be below. So the package that we use is called ProteoDA, and that is an R package, and it runs Lima, which Lima is a way of doing basically linear regression with really big data sets. And so what this R package does, and I'm not going to go through the R package. I didn't write it. I can use it, and I'd be happy to teach you how to use it. It's up on GitHub, but I'll tell you what it does. It separates the annotation from the intensities. So you take that output file, and it will do that separation in an R package. It will use the metadata file that you have created to assign the groups. It will filter out samples with missing values. It will filter based upon, one of the things that we like to filter on is you've got to have the value for that particular protein in at least 2/3 of the samples in one group, if that makes sense. So if you've got an N of 3, you've got to find that protein at least twice in one group for it to count it. Otherwise, it's going to filter that protein out. So if you just get 1 hit out of 5, and that's all you get, it's not going to count that protein at all as even it's not going to try to quantify that. The other thing that this ProteoDA does is it writes a normalization report, and we're going to talk about that. And then you can, this is kind of an interesting thing, but after it does this normalization, this package leveraged some software that people wrote, and it basically will normalize your data 7 different ways. And then we get to go and evaluate, look at it, set eyes on it. We do this for every project and decide which one of these normalization processes looks the best. And now sometimes normalization is not appropriate. Okay, for our IP experiments, we don't normalize because that doesn't. That's not what we need to do for there. Anyway. Then in the program, after we do that, then we go ahead and it writes the QC reports, and I'll talk about what those QC reports are. Then it adds the model design, and then it will compare the groups according to the contrast that you have indicated in your contrast file. Then it writes those Lima tables, which is basically the differential analysis. And then it writes the Lima plots, which are the volcano and the MD plots and all that. And this package was developed by people that were all in this group here a couple of years ago. So let's talk about normalization. So normalization is, and these graphs that I have out here to the side, I'm really just going to refer to these two. There's a lot of more graphs and metrics and stuff that come out through this package than what we really focus on. But in every week, we have a meeting and we go over every project that we have completed from the week before. And we go through all of these plots together and we look at them to decide if how the analysis went. So normalization, the data is normalized 7 different ways. The first one over there on the far left, this guy here, this is just the log2 transformed. And this, the idea is this is the, what we want to do is this, we want to try to minimize the amount of variation in this top plot, and we want to try to maximize the correlation in this bottom plot. So what we're, a lot of times, all these normalization techniques, you only have to look at a few dozen of these things to realize that a lot of times the normalization, these different methods that they have, median normalization and VSN and cyclic low S and all that. They all normalize basically about the same. There's some that'll do a little bit different, but they're all pretty much the same.
On a good data set.
On a good day. But most of our projects, if you look at them, are normalized either using VSN or cyclic low S. And I'm not going to go into the statistical nuances of how they do those, but those are two different packages that normalize. So this PVC plot, so which normalization provides the lowest variation? That's what we're looking for up here. If there was one that was extremely, a lot lower than the others. When you have data that's really good, a lot of times these plots look really boring. And then the COR plot is which normalization provides the highest correlation. And then when we look over at these plots over here, we're kind of seeing if the data is centered around that 0 point, okay? Is the data centered around the midline? Kind of gives us an idea about that. So from that, here's the other part of the normalization is it shows a graph of the log2 versus the mean intensity. So you got the log2 fold change on this side versus the mean intensity going here. And it's got a plot for each one of those normalization processes or the unnormalized, just the log2 transformed right here. And then it draws this orange line, which is essentially the best fit line representing the trend of the data. And so for certain data sets, big data sets, proteomic data sets where you are doing a treatment, your assumption is that out of the thousands and thousands of proteins that you're going to have, whatever treatment that you're going to do, there's only going to be a small subset that are actually going to do the changing. Okay, so the assumption is that most of the proteins are not going to change. And the other assumption is that whatever cumulative change effect can basically be summed to 0. So the amount upregulated and the amount downregulated. And so one of the normalization processes it's trying to do is it's essentially trying to get this orange line back horizontal to the 0. And so that's what these normalization processes are doing, is trying to adjust that data so that that best fit line then, but what that can do is you can see, especially if this orange line is really skewed, and especially if it crosses that midpoint line there, is when you normalize that, now sometimes you can have values that intensities that becomes negative. And they can do some kind of squirrely things. And so that causes a lot of consternation in people because they're like, I can't publish that. What does the negative value mean? You know? And so some people opt to just say, I'm not going to normalize at all. So choosing a normalization based upon this, you can see that the cyclic low S does a pretty good job here. And there's a whole science. You could have a whole class based upon just all of these techniques, but I'm gonna kind of go on and say, let's say we chose that, and then we go ahead and apply that, and we go ahead and look at some quality control plots here. So this is a violin plot, and this violin plot shows kind of a top-down version of the data collected for each sample. So you've got two different groups here. The blue group and the red group here are the two groups I'm going to compare. They're two different HeLa samples. And that line in the middle of it represents the normalized mean intensity. So you've got intensity along the y-axis here for each of the samples. And you'll notice that most of the data is Gaussian, and it's essentially found clustered around the midpoint. And then you've got these extremes where you have really a few highly abundant proteins and a few low abundant proteins that are here. And you're kind of looking at these tails to see, you know, in one of these samples, you can see like this guy here, the tail goes down a little bit farther here. You can see there's these are all really uniform along the top. Like I said, I worked in the resource in the first, I don't know, 3 or 4 months. Every time we would look at these plots and everything, and it's like they all look the same. And then one day we came up and there was a really bad data set. I'm like, whoa, they don't always look the same. So these guys will look like this if we do our protein quant on the front end correctly. But if we don't, then these...
Yeah, it can also just the protein distribution The initial protein distribution can be wrong. Solubilization can be wrong.
The solubilization can be up. Yeah, so so this is that's why we call these quality control, because they will indicate, it's kind of like you've got cars, they still have these little indicators, and that little red light comes on. My kids think you just ignore that because that's what mom does, right? You just ignore that light. Like, no, it's supposed to mean you're supposed to take the car in because something's wrong. So same thing, if there's something wrong with this quality control plot, something doesn't look right, then we need to address that. Yes, ma'am.
In that case, like one of the sample looks different from others, which means it is not normalized, right? So this normalization has to be carried out during the sample preparation or after the mass spec analysis. Where is this sort of normalization should be?
So, okay, so the question is this normalization here is all done on the data afterwards, but it is pointing to, essentially, you're right, we have done another normalization before the experiment began when we did our protein quant. Yeah.
We have not taken equal amount of protein, we may end up
You will be able to see it here.
we have taken equal amount of protein during the experimental time, and we may not get we may get equal normalized data, or still we get unnormalized data.
If you did the protein quant correctly on the front end, and like you said, we ended up solubilizing all the same amount of same types of proteins in our thing, then this will look it'll reflect this in this plot. So this, I guess the question that you're trying to address is this is just an indicator that all of the samples were essentially processed in a similar way, and that the material that was used. So if, for instance, I could show you if I had a different cell type in here, I didn't have a HeLa cell and something else was growing in there, because sometimes that happens, right? You end up with some sort of bacteria or contamination that grows in there, and you know what? You do a BCA, and you know what? Bacteria has protein in it, and so you think you've got lots of protein in there, and then when you do your analysis, it looks completely different because those aren't cells anymore. It's bacteria.
Or a cell lysate versus a serum sample. The violin plots look very different. Serum, there's a lot more low abundance proteins. It's not, serum is not normally distributed in terms of abundance. There's, you have a few really, really abundant proteins, and then you kind of an asymptotic tail down to low abundance. But cell lysate is more evenly distributed, and you see those distributions illustrated in the violin plots.
Yeah.
I have a question.
Yes, ma'am.
So when we do Western blot, we normally have like internal control, such as like GAPDH not changing, alpha tubulin.
Yeah.
So we normally, when we do our finalizer, we use that as a base. So is any proteomics has such a marker? No.
No.
Yes. So the question is that when you do a Western blot, you always normalize to some sort of housekeeping protein there. And do we do that in proteomics? And the short answer is no, we don't do that.
Yeah. So what are the base you normalize? Suppose I give you a sample A, same cell type.
Yeah.
Sample A, no treated. Sample B is treated.
Yeah. So that's what he's saying. We go through 7 different types of normalization. And so the one that makes sense to me is one we never use. It's the one I always used to use because it makes sense. And that's log2 normalized. And you take your data and you convert it to log2 intensity, and you take the median and you set the medians equal to one another. And that is a crude way of getting to a normalization. And so that's one I can wrap my head around. I can't explain to you the math behind VSN or cyclic low S. It's been explained to me and I understood it in that moment, and then I didn't understand it anymore. But it's a way to mathematically alter, it reminds me of calculus, really. And essentially what you're doing, you're just distributing the data in a different distribution, and then you're normalizing on something else. It's easy for me to visualize log2 median, but the others, you can see from those plots, the other methods do a better job of normalizing across the entire range of intensities.
Yeah, so follow-up question, I was wondering about the same idea. So now I understand that we're making the assumption that majority of the proteins do not change, and we're using this as a way to normalize it. So then.
Well, actually, not using that to normalize it, you're actually using that as an evaluation after you normalize, because you don't want the normalization to impart, that half the proteins have a twofold change. And if you saw that, then you would know, okay, the assumptions I'm making in the normalization are not correct, because I don't expect, a twofold change. And that's why it's difficult to normalize on a pull down, where you're pulling down with a scrambled vector, or you're, a target for your protein. You're going to get very different answers. It's difficult to take those proteins and normalize them because they're different, right? And the assumption isn't that they're the same with minor differences. There are huge differences.
So my follow-up question is, for pull down, you do not normalize.
We normalize, basically, if you're doing the pull down, we're hoping that you do a good job of normalizing the input. And then in those cases, and we didn't get into this yesterday, but oftentimes we'll ask for a sample of the inputs, because sometimes the inputs are different, and you may not realize. Let's say you're doing a pull down in two different cell types, a wild type and a knockout, and you're pulling out something. Well, the fact that you've got a wild type and a knockout may be changing the protein distribution in that group. And we actually had this occur at one point. In a pull down, we saw this family of proteins that were twofold upregulated. And it wasn't expected. They didn't expect that, that this would be interacting. And we went back and looked at the inputs, and those proteins were twofold upregulated in the input. And so what we were seeing was just a background of the, non-specific interaction of the proteins, mimicking their original concentration in those inputs.
So you also did a mass spec.
Yeah, we did a comparison of the inputs, yeah. And we recommend that. Everyone doesn't want to do it because it's, it adds cost, it adds samples. But especially if you're treating your inputs differently, if you're treating your cells differently, it's really a good idea to submit samples of your input material. But it's crucial that you have equal protein amounts, because that's the only normalization that we can do. Yeah.
So not just normalization, I think if your treatment is having a big impact on, let's say, low abundant or very long-lived or highly abundant proteins, you can see it from this kind of data, right?
You can.
So this is not just normalization tool, it's actually results.
Yeah.
Yes. Yeah. These we moved on to results here. This isn't just normalization. We did the normalization, and then we applied that, and these quality control plots here this is normalized intensities here. So to kind of sum up that conversation, I don't know how much you guys online are getting. It might be helpful if you would go ahead and log in to the Zoom and then turn your microphones on if you're going to talk, so that I just want to make sure they can hear.
We heard pretty well.
We could hear it.
Well? Okay, great.
Yeah, that came through pretty well.
I'm trying to yell.
Well, Rick is right here in my ear, so it's good.
Used to be yelling at him. So.
No, it's good.
Yeah.
Thank you guys for telling me that. All right, perfect. So yeah, when you and there are a lot of people that again, the normalization kind of makes people. There's a couple of things in bioinformatics that people kind of get a little nervous about. Normalization, one of them, and imputation is the other one. And imputation means where we magically make those NAs into a number so that you can get some log fold changes there, and everybody wants somebody else to make that decision and do that, and then they just get these. Anyway, we'll get to that bridge when we cross it, but. Cross that bridge when we get to it. All right, let's move on. So one of the other things in our quality control plot, and these are great. I'd love to be able to just continue, have you guys ask questions and talk about the things that are really interesting to you. The other important thing that we get is a principal component analysis of your data. And so, so each dot here in this plot represents a sample. And then, of course, we have two different sample groups. So there's two different colors here. And the x-axis down here is our first principal component. And the y-axis is that the next principal component 2. And the principal component 1 is the one that is, you can imagine if your data is, what variable would represent the most amount of variation in your data? That's going to be principal component 1, the largest variation. And then the second one would be principal component 2. Kevin, in his talk yesterday, had talked about principal component 3, which is a little hard to express 3 principal components on a 2-dimensional graph, but you can look at as many principal components as you want. But we usually just look at the first 2 to try to separate it out. And what you can see here is we get a 53% of the variation is in the first principal component and about 12% in the second. And so the way we think about that is like, even though this graph is a box, there's a lot more distance on the x-axis than there really is on the y-axis. So in other words, the difference between the groups is the variation between the groups is much bigger than the variation between these guys, because you have so much more variation in the y and the x direction here. So the farther apart the samples are in the separate groups, the more different the groups are. So what everybody, this is a result that shows that there is a difference between these two groups here. But inside, you've got some variability within the group. And you're not always going to get really, really tight clustered points together. And sometimes that's okay. It is what it is. Another way to represent this information is in a dendrogram, and the distance along the y-axis here before a branch indicates how different the samples are from each other. So the fact that you have this branching here and a long distance going down here before you start to see these little branches here, that just shows that there's a big difference between the two different groups, and there's a smaller difference within each group. And sometimes they'll have a project where you just don't have a lot of distinguishable, the groups are not so easily distinguishable. I mean, this is what happens when you have an analytical chemist design an experiment, right? I got some samples and I made it to where it's a no-brainer. Anybody could do this, right? But I'm also not going to publish this. A lot of times you guys are dealing with groups that are much closer together, and you've got a lot more variation in your samples because you're doing tissue from different animals or whatever. The other thing that we look at is called a similarity plot. And this plot just shows the correlation, or a correlation plot between the samples. So each one of these columns down is a sample, and it's the same sample here. So you see this big diagonal here. That's because you're essentially comparing the sample to itself, and so you're going to get the most correlation there, and the farther off axis that you get. And so what you can see is that the red group, essentially compared to the red group, is highly correlated, and the red group compared to the blue group is not so correlated. This Pearson correlation is much smaller for that. So that's, again, just one way of us being able to look at it. And what we're looking for, again, is there a sample in your group that sort of is sticking out, is an outlier, looks like it doesn't kind of belong with the rest of them? Those are the sorts of things we look at. Another thing in our report that we have that we provide with every project that we have here is a missing values plot. And so this only shows the missing values, which that means that the protein is absent in a sample, but is present in another sample. So you can see here we've got the white space means that we're missing a value here in this sample in the blue group here, but you can see that sample, that protein is in lots of the other groups here. And it kind of just gives you some idea of the missing values that you have and how many missing values that you have. And so if you look here, a lot of times you can see kind of a characteristic pattern that we have all of those proteins in this group. There's none of them are missing, but there's a lot of missing values here. So that could be that those proteins have been downregulated, and the difference between these groups. And so we're not seeing them as much. Yes, ma'am.
Does that mean that it is not detected in the mass spec, or it is not seen in the sample at all?
I would say that what we would say is that it wasn't detected. We cannot with 100% confidence say that that protein is not there. We can just say it's below we just didn't detect it. Well, number one is it could be because it's low level. So the changes between whatever treatment we are got going on here between the blue and the red has caused that protein, the peptide, the protein concentration is now lower. Therefore, there's fewer peptides. Therefore, there's less signal. And so the mass spectrometer is picking up on those, and it's now down in the noise, and it doesn't pick up on those things. And you got to remember that I'm going to use my hands here. So for those of you that are by Zoom, you're not going to be able to see this, but you know, you've got we do all this chromatography to squish and concentrate the peptides together into a sharp, narrow peak. And so if we have a low level peptide, that peak can still be narrow, but it's really short. So if I use the top of my computer screen, you know, you've got it when it's really concentrated, it's like this, but when it's really not concentrated, all you see is this little blip. And remember, the mass spectrometer is cycling through, and it's measuring, and then it's measuring, and it's measuring, but there's a cycle time as it's measuring. And so it may be that at a low concentration, that peak passes, and it doesn't, those ions don't get captured, they don't get filled, and they don't get analyzed because it's so low level. Yes, ma'am. Is there a cutoff for the peak intensity? I'm going to let Rick answer that one.
There is, but there's not a hard and fast rule that we say any peak below this we're saying is not detected, because it depends on what the background is at that time. There are regions of the spectra, you know, regions of a chromatogram where there just aren't many peptides. And because there's just not many peptides, you can actually see a very, very small signal. If that small signal were present 10 minutes earlier in the chromatogram, there's just too many peptides that are really not quite at the detection limit, but the summed intensity of those causes more noise in the spectrum. And so it's more of a signal to noise issue than signal issue. But it's a moving target, and it depends on the charge state of the peptide. And so, you know, there's about probably 6 different factors that come into play, but we don't use a hard and fast. If it's below this level, we're not going to look at it.
It's kind of relative to.
And it is, and it's relative to each instrument as well.
Yeah.
Good question.
There are certain proteins which are high abundant in sample, but they don't get detected in the mass spec.
Sure.
So
Sure.
what are the
I always blame glycosylation. I blame glycosylation. And so there are certain proteins that don't digest well by trypsin. One are glycopeptides, proteoglycans more specifically. So a proteoglycan has more sugar on it than protein. So it may be there may be 15 or 150 residues of amino acid, and it should be 15,000 molecular weight, but instead it runs on a gel at 80,000. And this is because it has lots of big glycosylation, big sugar groups on it. And so these can mask the protein and keep it from being cleaved. I ran into this with a clinical researcher who didn't accept that as an answer. We were working with multiple myeloma and these patient-derived samples that we were isolating based on the presence of CD138. CD138, another name for it is syndicans. It's a protein that's expressed on multiple myeloma cells, and so we had an antibody-based method. So, you know, the protein had to be there in order for us to select it. 95% of the time, we couldn't detect that protein in the cell lysate. And this is a few years ago, we were probably detecting 4,000 proteins. And that wasn't one of them. It was one of the more abundant proteins. But it was so small in terms of protein molecular weight and so big because it's a surface protein, proteoglycan, lots of big sugars that trypsin just didn't do a good job of cleaving it. And so we rarely, if ever, saw it. And the doctor just couldn't quite accept that as an answer. It was the answer, but he didn't like that. So I blame Kyle.
Mike had a comment too.
Oh, I just pointed out that some integral membrane proteins and some of the small mitochondrial proteins have similar problems, that when you digest them with trypsin, they just don't make very many good LCMS friendly peptides. So they can just be effectively invisible to the experiment.
Yep. That's right. So one of the things that Mike does in his targeting thing is he spends a lot of time when people say, I want to look at this protein, and he says, well, let's see what peptides we can actually see. And so then he's looking at, and in his talk, he'll talk about best flyers, and because it's not just about concentration, it's about having peptides of a certain size, of a certain makeup, and again that are not masked by other PTMs that will allow you to be able to see it.
So,
This plot for, like, let's say IP experiments.
Mm-hmm.
In IP experiment, I mean, the goal could be to find identify proteins that are modified only in one group. So then can this kind of plot tell you?
Yeah, we're going to talk about that. So I'm going to show you, because a lot of times in a volcano plot is not very helpful for an IP experiment, because it's really presence absence. And so you are going to see blocks like that. And so there's a way in which, when you have the differential analysis worksheet, you can sort that, you can filter that, so you can look for only those that they are basically all NAs in one group. And so on a volcano plot, those all show up, basically it's zero. They're not there because the group doesn't exist. It excludes all those. But that presence absence, and that's something we spend a lot of time trying to coax people into, because they're like, well, just put a value in there. I just want a number so that I can get a full change. I'm like, you've got better than a full change. You can say, we have a sample here where we verifiably measured it, and now we have a sample where it's not there anymore. I mean, what's better than that?
So regarding the glycosylation mask during the sites for digestion, is there any strategy to solve that problem?
Mm-hmm.
Yeah, you can treat with a deglycosidase, so you can remove some of the sugar with just a non-specific glycosidase, and then subject it to digestion.
Yep. Great questions. Good stuff. Okay. And then the last thing is, our missing value plots sometimes can allow us to identify an outlier sample. You have one sample in your group that just has a lot of missing values. And so you're kind of like, yeah. So we're going to move on. And so after you do that, we look at the quality control, then we look at the differential abundance. This is where we're going to actually look over the data. And so we're going to look at, I'm just going to kind of tell you what we're about to do, and then I'm going to pull the slides up and we're going to look at it together. But we're going to look at a volcano plot. That's where everybody wants to go. We're going to be able to show you how you can figure out the number of protein IDs, the depth at which that experiment went. Then we're going to sort the data. With that volcano plot, there's a way where we can look at sort it by intensity, log full to full change, p-values. You can do this in the table or you can do this in the plot. And then show you there's also a way to go from a volcano plot to an MD plot, which basically adds the dimension of intensity so that, because sometimes when you're looking and you're like, hey, this looks like a really significant protein, but then you say, well, why? You don't get a sense of what the intensity is of that protein like you do on an MD plot. And then I'll talk about those presence absence comparisons and show you that Excel table there. So let me back out of here and open this guy up. Okay, screen sharing is shown. Okay, that's right. We're going to share again here. So let me go back over here. Okay. So can you guys see that? Am I sharing the right screen here?
Yes, we can see it.
Okay, so just to kind of go through, so part of the output that we do is provide you with this. This is not a website. This is an HTML document that is included in the folder that you get that is basically tied back to the table. So as long as you keep the table with your differential analysis in the same place as this, the pointer should point back to it, and you should be able to continue to use this in any browser. It doesn't have to be Chrome or Edge or whatever you want. So there's your volcano plot here, and I just want to go through. So first of all, remember we talked about in your contrasts what the title of this was. This is where you have declared that we are taking group B and we're subtracting from it the values of group A, because, and maybe, I mean, surely we know this by now, but all of the quantitation that we do in our global proteomics is all relative. What Mike is going to talk about in the targeting section, that is absolute quantification, where he can tell you concentrations. We can tell you that this value is so many fold changes above this other value here. And so that's what the volcano plot is basically showing you here. So we have the volcano plot here, and then any one of these points that you have here, each one of these dots represents a protein here. And if you hover over it, it gives you some of that same information that you'll have in the intent. And then so here's a good example of one where we have really high intensity for this guy in this group, and the intensity is really low in the other group, and we have a lot of missing values. So we are only getting one data point out of the 5 for this other group here. And so that's because group A is much higher than group B here, and the way that we have this compared, this would be downregulated. And then once I click on that, it pulls up that exact gene symbol and the description and the average intensity, and all that information is down here. And if you scroll down a little bit farther, it will tell you how many proteins. It says showing 1 of 1 entries found from 2,139 total entries. So that's telling you what the depth was for this particular experiment. And I think that is because the reason why this is so low level is because I think I was only injecting 100 or 200 nanograms of our JJN3 here, and then looking at that. So this is your volcano plot. And so the idea, the strategy would be, as we tell people, is that, to find the candidates. So the candidates would be starting from these corners and sort of moving in. You would, obviously pick off the low-hanging fruits, all of these guys that are in red or that are blue. But there may be some other ones in here that are right along that line. You can see there's kind of an invisible line here, and there's some of these guys that are just on the other side of it that might be part of your biology that you're looking for. So the other thing that we can do is, again, you can sort it by intensity. You can look at all the high intensity guys first. Of course, actin's the top of the list. That's really exciting. Everybody wants to do studies on actin these days. Histone H4 is always a really abundant guy. And then you could also look at the log, which ones have the greatest log fold changes, adjusted p-values. This is the one where I was talking about when you look at this one with the adjusted p-value and it doesn't have a value at all, it's because this is the presence absence thing. So we don't even have any of the protein measured in the other group, but we have it measured all 5 times in that group. The other thing you can do is you can go over here and look at the MD plot. And so this is the MD plot. And you guys were asking about, I think over here about intensities, like kind of what the cutoff was. And you can kind of see the natural cutoff of where, you know, for the average intensities, it gets down to a certain level. Certain instruments that we have, this level gets even lower. So sometimes we artificially filter it out and we say, okay, once it gets below a certain level, there isn't anything good down there anyway, so we're going to just cut that off. And sometimes the instrument does that for us because its sensitivity drops off there. And so, you know, the highest abundant guys are over here and the lowest guys are over there. And so this is one way to interact with your data. And this is a tool that was developed as part of this ProteoDA package, and we use this all the time. The other part that is, let's see here. I want to make sure that when I share the screen, are you guys able to see the spreadsheet now, or do I need to go?
No, I don't see a spreadsheet.
Okay. I think I have to share. I'm sure I'm doing this wrong, but okay. So then the other thing that we get is this, what we call the differential analysis worksheet here. And so again, we put all that back together again. We've got your, over here, you got your protein annotation, and then these are all filterable. So you can filter by these guys differently. It's got it all broken down. These, the Uniprot IDs, if I click on this guy, it'll take me actually back to the Uniprot site where you can look up that particular protein. So each one of these rows here represents one of those proteins. And then the next group here, this is going to be the normalized intensities, and it even tells you what type of normalization. So we did log2 transformed and then the cyclic low S normalization for all. In this case, there are this is a subset. This is just 10 samples. This is just the JJN3, which is I guess it's a lung cancer cell line, myeloma cell line comparison between these two groups. And so you can see that it's got all those normalized intensities here. Remember, I told you before, there's a TSV file that is the unnormalized results from your database search. This is the normalized after you've gone through this bioinformatic process here. And so you can see that these, instead of NANs, as it comes out of Spectronaut, these guys are changed to NAs. So we'll have all of that data here. Those are the intensities. And then whatever comparison. So we only had one contrast here, just the two groups compared to each other. So we'll have just one set of statistical values for that, starting with the log fold change, and then you've got the confidence intervals and the average intensity, and then you've got some statistical parameters here, and then you got your p-value and your adjusted p-value. And again, we can, these are all filterable, so I should be able to come in here and say, let's see, now it's going to show me only the ones that are upregulated and downregulated and that have a significant p-value. And so then I could take this group of proteins here. You can see it's not all of them anymore. It's selectively pulled those out. And if you want to do some pathway analysis or you want to make a plot or something like that, you can just go grab these guys and do all kinds of great stuff with it here. And then the other thing we can do is if I come here and I these guys again, and I told you that. So if you come and you look at The NAs. Yeah. So for this particular data set, there was not a lot of proteins that were present absent. But these are the 4 that we have all values in all of the of one group and none of the other. And so you can see that we've got 3 of them that are all in the first group, none in the second group, and then one of them that's in the second group and none in the first group here. And so that would be a way to be able to sort your data and quickly be able to look at those. Now, you know, and I'm looking at these and I'm like, okay, these proteins, this is not my biology, I don't know, you know, whatever, but you might look at those and go, oh, those are exactly the ones I'm looking for. And so that's that would be a great value to you. And so we try to provide you with some tools here where you can, from the samples that you sent us, be able to get to the place where you can make biological decisions. You know, you can look through the quality control data and kind of make your own decision on how confident you are of how well the experiment went, because really you're sending off samples and you're sitting there waiting by the mailbox for these graphs and things to come back. But you know, you don't know if like instruments were on fire, we had a flood in the lab, you have no idea what, you know, what happened there. But when you see all these quality control missing value plots, all of these things, you can kind of piece together, okay, I have confidence in this. Looks like, you know, the experiment went well on my end, the sample prep went well. You know, you can even open up we even send you the raw files, and so you could even look at those, and I can show you some other ways in which we, you know, it's probably a little bit outside of what you guys do, but show you how we also look at file size as we're collecting the data. They tend to all be about the same size. And if you have one that's dramatically different, you know, that usually means that we had maybe an injection error, right? It's a bad word, but we get air bubbles from sometimes. And so it's not injecting your sample, it's injecting air. And so we have to go back and we evaluate all of that. Because by the time you get to this place where you're making your biological decisions on this, you want to feel confident that that data is firm and sound. And so we want to provide you with all of that, because really you don't want to go chasing down a rabbit hole that really isn't there. Okay.